go right on just to stay on schedule. And Zach Hornby from Ignyta will give a presentation about a very innovative basket clinical trial called Star Trek II.
So good morning. I'm Zach Hornby from Ignyta. I enjoyed this morning's session because it helped frame the high-level issues that are the key considerations for genomic-driven trials. And as you just heard from Roy, when it comes to oncology studies, there's two key frameworks that you can employ for these studies. And so Roy just talked us through an umbrella study, which is a single histology, in this case squamous lung, with multiple different markers, and for each marker, a different drug. My talk is going to be on kind of an inverse trial design, which is called a basket study. And in this case, there's multiple histologies, so all solid tumor histologies, but a very small subset of markers, and then just a single drug. So one drug, multiple histologies. Before I dive into the trial design and the lessons learned, I'd like to take a moment to tell you about our company, Ignyta, because it may be less familiar to this audience. We're a San Diego-based precision medicine oncology company, a public company founded in 2011. We're developing a small pipeline of targeted oncology agents. The lead program, entrectinib, is what I'll talk about today, but that's also followed by several other targeted agents. What makes Ignyta unique as a company is that not only are we developing target oncology drugs, but we're also developing and performing in-house the companion diagnostics that help select the appropriate patients for treatments with our therapies. And so that makes us somewhat unique as a biotechnology company to be doing both diagnostics and drugs in tandem, but we think that's critical to really understanding appropriate use of our drugs. Our lead program, entrectinib, is an inhibitor of 5 protein targets, the TRK family of proteins, ROS, and ALK. Most of you are probably familiar with ALK, if not ROS, but TRK is a novel target. Interestingly, all 5 of these proteins are cell surface receptors that are prone to genetic translocations that lead to fusion proteins that are constitutively activated, and they activate oncogenic downstream signaling pathways such as MAP kinase, PI3 kinase, and PLC gamma. And so when those pathways are activated, these cells hyperproliferate and survive. And so these are very strong oncodrivers when these fusion proteins are present. Our compound, entrectinib, is the most potent TRK and ROS inhibitor in clinical development. And when applied to these fusion protein driven cells, it does indeed downregulate the downstream signaling. Importantly, this compound was also designed to cross the blood-brain barrier, which is now becoming a key consideration for oncology drug development. So when we first started working on this program, we wanted to seek to understand what are the tumor types that are driven by this mechanism, and therefore how should we design a clinical trial to best study this. And so we obtained data from sources like TCGA, the AACR, Project Genie, Foundation Medicine, and even literature searches in PubMed. And what you can see here is that each of the TRK, ROS, and ALK fusions are found across a host of different histologies. However, within any given histology, they are quite rare, generally on the low single-digit percentage of prevalence in large tumors such as colorectal or lung cancer. Interestingly, in very rare tumors, these fusions can be quite common, if not pathogenomic. And so that applies to certain subsets of cancer like mammary analog secretory carcinoma, which is a salivary subtype, or secretory breast cancer. But through our research, we've identified more than 30 different tumor types driven by TRK fusion proteins in specific. What's also critical is that our compound is active irrespective of the fusion partner, and more importantly, irrespective of the tissue of origin. So you can see here a representative list of about a dozen or so preclinical models, each representing different tumor types, such as head and neck cancer, melanoma, colorectal cancer, et cetera. But what you can see is that the drug maintains single-digit nanomolar potency irrespective of the model system. More importantly, this mechanistic observation was borne out in the clinic in the phase 1 setting. What you see here are the results from 2 phase 1 studies. These data were recently published in Cancer Discovery last month. And you can see that our agent entrectinib had a 79% objective response rate in the phase 1 setting in patients who were harboring these fusions. Importantly, that response rate was consistent across each of the molecular targets, TRK, ROS, and ALK. And what also is represented here is 6 different tumor histologies in which these responses were observed. So this is not a drug that's specific just for one cancer type, but rather multiple cancer types so long as they have the relevant mechanism. So what we know is that the target is real and we have an active drug, but herein lies the challenge for drug development is how do we design a registration-enabling clinical program and the companion diagnostic approach that finds and enrolls these patients that while within a given tumor histology are relatively rare, collectively they're quite numerous. Aspects to consider are many of the tumor types that would be relevant for enrollment don't currently participate in genetic testing. And even when genetic testing does occur, some of our biomarkers of interest, such as NTRK, may not be on the standard panels. So this slide captures the solution that we employed. It's an overall flexible trial design. As I mentioned, it's called a basket study. So it's open to all solid tumor patients as well as lymphomas, so long as the fusion proteins are present. We're employing a pooled statistical analysis such that each individual tumor histology collectively contributes to the overall primary endpoint. We allow multiple different mechanisms of diagnostic detection. One of those is our central lab at Ignyta, and I'll tell you a little bit more about that. But we also allow various regional or national commercial diagnostic labs and even local academic labs to detect the patients who are eligible for the study. We have very liberal enrollment criteria, which again, I'll tell you more about. We have a pretty neat mechanism called just-in-site trial activation where we can open up trial sites throughout the country when a patient is found, but not before that. And then finally, we are working with patient advocacy groups to raise awareness for the study and importantly for the need for genetic testing. So this slide is a schematic of the trial design itself. I'll just point out a couple things. The first is that, as I mentioned before, all solid tumor histologies or lymphomas are eligible so long as one of these fusions is present. And then the other important consideration is that the molecular testing may occur. And as Roy mentioned in his talk, we allow pre-screening or screening at any point in a patient's care. So they can be responding to another drug and still undergo the genetic screening. We enroll patients everywhere from first line to multiple lines. We've got patients 7th line on study. Also importantly, we have a companion pediatric study that mimics the same design and largely mimics the same trial sites. This study was designed in collaboration with the Children's Hospital of Philadelphia, as well as a patient advocacy group called the Alex's Lemonade Stand Fund. And again, we're enrolling fusion patients because the mechanism is not only agnostic to what tumor histology it resides in, it's also agnostic to the age of the patient. So this is an important consideration that our scientific hypothesis is any patient harboring one of these fusion drivers should be eligible for study. So as I mentioned before, the way that this basket design is enabling is that each basket, which is defined by the presence of a specific fusion and a specific histology, can all contribute collectively to the primary endpoint, which is objective response rate. And what this might ultimately yield in collaboration with the regulatory agencies is potentially an unprecedented label indication, which is what we call a molecular label, or an indication statement that is not defined by the tumor histology, but rather by the mechanism alone. So the goal is to have an agent that is approved for NTRK fusion positive cancers, hard stop. As I mentioned before, one of the key challenges is finding these relatively rare patients for enrollment. We employ what we call our Trident strategy. It's a 3-pronged approach. So the central tenet of this strategy is that we do have a central assay in our lab at Ignyta. It's called the Trailblaze Pharos assay, and I'll tell you a little bit more about it. But this is in our CLIA and CAP accredited lab, and ultimately this assay will be the basis for a PMA, which is requisite to be applied for in parallel with an NDA for a targeted therapy. In addition, we're working with various diagnostic and diagnostic consortia. These can be pan-national initiatives such as the EORT SPECTA initiative in Europe, but also commercial labs such as a Foundation Medicine or Garden or Caris here in the US. And then finally, our diagnostic team from Ignyta actually travels to various academic sites throughout the world, from Asia to Europe to the US, helping enable certain sites that have next-generation sequencing capability to add our relevant biomarkers to their panels so that they too can detect these patients locally. This is a depiction of our assay at Ignyta. So I mentioned we have a CLIA and CAP accredited lab. Our assay, Trailblaze Pharos, was designed specifically to test for these rare genetic fusions. We designed the assay in collaboration with CDRH. We have been granted an IDE for this assay. You heard in one of the earlier panels that that's an important consideration. We also have been granted FDA's relatively new EAP designation, which is for diagnostic assays that serve unmet needs. And so any patients detected at our own lab then become eligible for our study. Once you find a patient, the challenge or the battle is still not over. You need to be able to determine whether in fact they're eligible for the study based on inclusion-exclusion criteria. So this slide was actually generated for the Friends of Cancer Research annual meeting last year, where the first session in November was focused on how do we widen eligibility criteria to get more patients on study. And so the general premise that we've taken is that we want to allow very broad enrollment. We understand that we need to protect the integrity of the primary intent to treat population and primary endpoint, but we may be able to also, through other baskets, generate real-world data that help inform the real-world use. And so how we apply that specifically for our trial is that we have broad inclusion-exclusion criteria, as I mentioned earlier, across different age ranges and histologies. We don't have very restrictive criteria when it comes to things like renal function, hematological function, et cetera. We do allow patients with CNS disease. And then finally, we do have a basket for patients who won't contribute to the primary endpoint. For example, these may be patients with non-measurable disease or very poor performance status. Another key aspect to this study is because we need to find these rare patients, we've gone global in design with a catchment area of 14 countries worldwide on 4 continents. We've got more than 150 clinical sites, including 70 here in the US, an additional 150 that can be activated just in time based on a central IRB and central contracting process. And then finally, we will kind of white glove travel patients from anywhere in the world who are not near a clinical site. Importantly, for raising awareness are the initiatives we have working with patient advocacy groups. These are a couple representative examples like Sarcoma Alliance, Inspire, American Head and Neck Society. We try to present at conferences and share live data updates that are specific to those therapeutic areas. We also participate in newsletters, banner ads, e-blasts. Importantly, these aren't just advertisements for our trial, but are more specific messaging around what are some of the known markers in the tumor type or encouraging genetic testing. Then we've performed search engine optimization for patients who are now becoming more educated when they receive, for instance, a Foundation Medicine report telling them they have a certain alteration. Of course, they're going to look it up online, so we want to make sure they become aware of our study. And finally, we do have a trial website, one designed for providers and another designed for patients. So I'd like to conclude by providing 2 case studies of how these mechanisms have helped us identify patients. The first one is a case where one of our commercial diagnostic partners identified a potentially relevant patient for us. This was a ROS1 positive non-small cell lung cancer patient. So on day 1, we contacted that patient's provider and asked whether the patient would be interested in a clinical study. Simultaneously, we looked at our just-in-time site network to find the closest proximal site for the patient, and we reached out to that site to see whether they'd be interested in opening the study on behalf of the patient. By day 4, via the central IRB, we had that site approved for the clinical protocol. By day 5, we had drug at the site and a fully executed contract in place. By day 6, the patient was visiting the provider, signing consent, and screening for enrollment. And by day 7, from the patient's detection, that patient was on trial receiving drug. So this shows you the power of this collaborative network of diagnostic screening partnerships and just-in-time site activation. My final case study is one that actually falls outside the framework of the clinical trial itself. This is a case of a 20-month-old baby who was born with a very rare congenital fibrosarcoma, who unfortunately had amputation of the leg below the knee at birth, later had metastases to the lung, had a year of chemotherapy. Unfortunately, the disease came back, now bulky disease in the brain. The patient had salvage chemotherapy to no effect. And at that time, we had a pediatric study ongoing, but we actually didn't have a pediatric formulation yet. We had only an adult capsule formulation. Another company had a similar agent and did have a pediatric formulation available, but they actually turned away this patient for their study because they didn't want to pursue a patient with bulky CNS disease. So our company, we worked with the FDA to design a single patient protocol, compassionate use, where we literally took our capsules, broke them open, and dumped the drug into yogurt. One of our employees, an older gentleman with no children, actually went and tasted about 20 different baby foods and assessed compatibility to make sure that we could come up with an admixture that would work for this patient. And so the good news is we were able to get this patient on drug. And what you can see at baseline is the patient had a major cystic lesion in the brain, significant edema, and you can see the shift of the midline within the brain. And that's at baseline. Patient was minimally responsive, not eating, really not crawling around. So you can see at day 35 on drug through this compassionate use protocol, you can see the reduction in the lesion size, the reduction in the swelling, the normalization of the brain midline, and importantly, the patient was now eating, mobile, more alert. So this shows you the power of being flexible and enrolling patients, sometimes not just to your trial design. So I'll close with my lessons learned. Rare patient populations that are highly actionable are worth pursuing despite their rarity. You should design your clinical trial to match the biology and the mechanism that your drug targets. You can engage the regulatory agencies very early. We've found that they're significantly more open-minded than we may have thought a priori. You should be open to building rather than buying when a ready-made solution doesn't exist. So the specific application here is that we built our own diagnostic assay early on when there were no other satisfactory diagnostic assays. But as the field has evolved around us, we're now collaborating with the diagnostic companies. And finally, and most importantly for our team and internally, compassionate use cases may not drive the primary endpoint, but they sure do invigorate the team and the company. Thank you.